Hello children, let's continue our class by explaining Calvin cycle now. Calvin and his co-workers worked out the whole pathway and showed that the pathway operated in a cyclic manner. The RUBP was regenerated. Okay, so this pathway is having 4 steps. Carboxylation, carbon fixation is the first step. So carboxylation is the fixation of carbon dioxide into a stable organic intermediate. So the carboxylation is the most crucial step of the Calvin cycle, where CO2 is utilized for the carboxylation of RUBP. This reaction is catalyzed by the enzyme RUBP carboxylase, which results in the formation of 2 molecules of 3-phosphoglyceric acid. Since this enzyme also has an oxygenation activity, it would be more correct to call it RUBP carboxylase oxygenase. Okay, wait, I will show you the structure. The cycle. Then in the second step, reduction. There are a series of reactions that led to the formation of glucose. This step involved utilization of 2 molecules of ATP for phosphorylation and 2 NADPH for reduction per carbon dioxide molecule fixed. The fixation of 6 molecules of carbon dioxide and 6 turns of the cycle are required for the formation of 1 molecule of glucose from the pathway. Okay, the next one, regeneration of RUBP. Again, regeneration of the carbon dioxide acceptor molecule RUBP is crucial if the cycle is to continue uninterrupted. The regeneration steps requires 1 ATP for phosphorylation to form RUBP. Hence, for every carbon dioxide molecule entering the Calvin cycle, 3 molecules of ATP and 2 NADPH are required. Okay, so it is probably to meet this difference in number of ATP and NADPH used in the dark reaction that the cyclic phosphorylation takes place. Okay, then about the C4 cycle. Children, plants that are adapted to dry tropical regions have the C4 pathway. Okay, so they are having a specialized anatomy which is known as Kranz anatomy, where the bundle sheath cells, they are following Kranz anatomy. Kranz means wreath and it's a reflection of arrangement of cells. So the bundle sheath cells may form several layers around the vascular bundle. Okay, and it is seen among the plants which is seen in dry arid region. Okay, so see what is happening in mesophyll cells. The first one, the fixation, carbon dioxide fixation is happening, and the first formed one is C4 acid. Okay, so that's why it is called C4 pathway. So the primary CO2 acceptor is 3-carbon molecule PEP and is present in the mesophyll cell, and the enzyme responsible for the fixation is phosphoenol pyruvate, and the C4 acid. The C4 acid formed here is OAA, that is oxaloacetic acid. Okay, it is then forms the other 4-carbon compound like malic acid or aspartic acid in the mesophyll cell. So this is mesophyll cells. Then it is transported to bundle sheath cells. So in bundle sheath cell, the C4 acids are broken to CO2 and C3. See, observe these steps, children. Then again the C3 transport again back to that C3 again back to the mesophyll, where it is converted to PEP, and other steps continuously occurring. Okay, so the CO2 released in the bundle sheath cells enters the C3 or the Calvin pathway, a pathway common to all plants. So the bundle sheath cells are rich in an enzyme ribulose biphosphate carboxylase oxygenase, RUBISEO, but lack PPase. Thus, a basic pathway that results in the formation of the sugars, the Calvin pathway, is common to C3 and C4 pathway. Okay, and next one, photophosphorylation. photorespiration. What is the difference between this photosynthesis and photorespiration? Why these two smileys are given like this? Children, to understand photorespiration, you have to know more about the first step of Calvin pathway. What is that? RUBP plus carbon dioxide gives 2 into 3-phosphoglyceric acid, isn't it? So this Rubisco, that is the most abundant enzyme in the world, is characterized by the fact that its active site can bind to both CO2 and O2. Okay, hence the name. So can you think how this could be possible? Rubisco has a much greater affinity for carbon dioxide when CO2 is to O2 is nearly equal. So children, imagine what would happen if this were not so. This binding is competitive. It is a relative concentration of oxygen and carbon dioxide that determines which of the two will bind to the coenzyme. Clear? So it will be a wasteful process. Okay, that's why this smiley. Clear? So in C3 plant, some oxygen does bind to Rubisco and hence carbon dioxide fixation decreased. Here the RUBP, instead of being converted to 2 molecules of phosphoglyceric acid, bind with oxygen to form 1 molecule of phosphoglycerate and phosphoglycolate in a pathway called photorespiration. So what happens, in the photorespiratory pathway, there is neither synthesis of sugar nor ATP. Okay, you have to underline that sentence. In the photorespiratory pathway, there is neither synthesis of sugar nor the ATP. Rather, it results in the release of carbon dioxide with the utilization of ATP. So, in the photorespiratory pathway, there is no synthesis of ATP or NADPH. The biological function of photorespiration is not known yet. Okay, so that's about the lesson, children. So here now we are completing class +1 biology. I hope all the topics are clear. Children, you have to read a lot because that much topics are there. So you have to read a lot, study well. Okay, thank you, children.